A new non-invasive method for measuring brain blood flow with light has been developed by biomedical engineers and neurologists at the University of California, Davis, and used to detect brain activation. The new method, functional interferometric diffusing wave spectroscopy or FIDWS, promises to be cheaper than existing technology and could be used for assessing brain injuries or in neuroscience research. The work is published in the journal Science Advances. The human brain makes up 2% of our body weight but takes 15 to 20% of blood flow from the heart. Measuring cerebral blood flow is important for diagnosing strokes and for predicting secondary damage in suprachoroidal hemorrhages or traumatic brain injuries. Doctors who provide neurological intensive care would also like to monitor a patient's recovery by imaging brain blood flow and oxygenation. Existing technology is expensive and cannot be applied continuously or at the bedside. For example, current techniques to image cerebral blood flow require expensive MRI or computed tomography scanners. There are light-based technologies such as near-infrared spectroscopy, but these also have drawbacks in accuracy. The new method takes advantage of the fact that near-infrared light can penetrate through body tissues. If you shine a near-infrared laser on someone's forehead, the light will be scattered many times by tissue, including blood cells. By picking up the fluctuation signal of the light that finds its way back out of the skull and scalp, we can get information about blood flow inside the brain. Naturally, that signal is extremely weak. The researchers overcame that problem by making use of interferometry, the ability of light waves to superimpose, reinforcing or cancelling one another. In particular, through interferometry, a strong light wave can boost a weak light wave by increasing its detected energy. They first split the laser beam into sample and reference paths. The sample beam goes into the patient's head and the reference beam is routed so that it reconnects with the sample beam before going to the detector. Through interferometry, the stronger reference beam boosts the weak sample signal. This allowed the team to measure the output with a type of light detecting chip found in digital cameras instead of expensive photon counting detectors. They then use software to calculate a blood flow index for different locations in the brain. The researchers found that with this new technology, they could measure blood flow more rapidly and deeper below the surface than with current light-based technology. They could measure pulsating cerebral blood flow and could also detect changes when volunteers were given a mild increase in carbon dioxide. When volunteers were given a simple math problem, the researchers were able to measure activation of prefrontal cortex through the forehead.